All right, here I am again, your Professor Carver, and today we are going to talk about one of the most important system in our body, the urinary system. It's very important because if you become a doctor and your patient came with kidney failure, you understand what's happened to this poor patient. So you can give him the proper treatment for it. So the urinary system, he play a role as this internal regulations that we call homeostatic. He regulates the blood volume and the blood pressure by adjusting the volume of the water lost in the urine, and release erythropoietin and renin, regulate plasma ion concentrations. He helps stabilize the blood pH. He conserves important nutrients. Assists also the liver in her function as detoxificating organ. And also he is responsible to remove inorganic waste from the body fluid. So he's made of two kidneys that form the urine, and each kidney so have an ureter that passes the urine to the urinary bladder, and the urinary bladder, this is where temporarily the urine is stored over there. So the urine is produced by the kidney, go through the ureter, go to the bladder, store over there temporarily, and then the urethra will convey this urine to the exterior. So we have a left and right kidney, and as you can see, the left kidney is seated up superior to the right kidney, right? And each kidney have a supply for renal artery and vein arteries. They are branched from the inferior vena cava, and they enter those arteries. They enter to the kidney through the hilum, this narrow passage called hilum. This is where also the ureter that will conduct the urine is getting out from the kidney. Reaching the urinary bladder. So let's look at the histology. Frontal sections through this is a left kidney. So it's made of renal cortex. First, it's covered by fibrous capsule, right? And we have this renal cortex. This renal cortex is the superficial portion of the kidney in contact with the renal capsules. It's reddish brown and granular. This form over here, it's called like, it's a form like a triangle, right? A pyramid. We call it a renal pyramid. We have 6 to 18 conical or triangular structures of this renal pyramid, and they exist in the renal medulla. That's the renal medulla, cortex medulla. Within the medulla, we have those renal pyramid. We have 6 to 18 distinct triangular structure. The base is about the cortex, that's the base, the pyramid. And the tip, this is with the renal papilla project into the renal sinus. Renal sinus is this internal renal cavities which lined by fibrous renal capsules and it plays a role on stabilizing the ureter position. And also the blood vessels and nerves. We can see here some blood vessels, red and blue. Minor calyx. That's the minor calyx, and then this is the major calyx. Is connected to the minor calyx through the connections. This is the connection. Renal pyramid. And renal cortex. And this is the renal column. The renal column is the space, actually, it's a band of cortical tissue that separate adjacent, separate each pyramid together. That's a renal column. So parts of the renal column, the cortex and the pyramid, they form a kidney lobe. So this is one kidney lobe. So one kidney lobe, it's a renal pyramid, this triangular structure, renal part of the renal cortex, right? And renal column. So this is one renal column over here, renal column over here, renal column over here. Renal pelvis, this is a funnel type shape chamber, consists of 2 or 3 major. Here I have 3, 1, 2, and 3. Consists of 2 to 3 major calyces. And it's connected to the ureter. يد hilum. Again, another structure. Sorry, I went too fast. So the renal pyramid, colons, renal cortex. This is renal medulla, the major calyx, minor calyx, renal papilla, the hilum, the ureter, and this is one kidney lobe. This is another kidney lobe. This is another kidney lobe. And all covered by fibrous capsules. So let's look at the blood supply for the kidney. It starts with the arteries. We have the segmental artery that is branched from the renal artery So he received blood from the renal artery, and he got divided into interlobal artery. He go into the lobes. Remember those lobes? This is one kidney lobe. This is another kidney lobe. Okay, interlobal arteries. Which radiate outward through renal canal between renal pyramids and supply blood into the arcuate arteries. Arcuate arteries, those. For some reason, I don't see the arcuate arteries. Oh, legend. I have it over here. So yeah, that's the one that's arch over here. Okay. So they do arch along the boundary between the cortex and the medulla of the kidney, right? Between the cortex and the medulla of the kidney. The arch form like an arch, all right? We have some arterioles that are going to branch from those interlobal. These interlobal arteries are going to be branched not only into those cortical radiate artery that they are like an arch, but also into these cortical radiate arteries. See them, those little teeny things, they are branching. And those are going to deliver, those cortical radiate arteries are going to deliver blood to the capillary supplying individual nephron. And we will go back to what is nephron, what is this, the functional unit of the kidney. It's called nephron. We will see it. Just give me a few seconds and we will be there. And those are going to be, since they are entering to deliver the blood to the capillary supplying individual nephron, they are afferent arterioles. All right. We have also the vein, interlobal veins, that they are coming from the renal supply from the renal veins. They also branched into interlobular veins, right? That they branch into arcuate veins. So in another word, I mean, the arcuate veins, they are going to empty into the interlobal vein, right? And then those interlobal veins will empty in the renal veins, will drain directly into the renal veins, all right? So the cortical radiate arteries that supply the capillaries that supply each nephron, they bring the blood from the renal artery, right? Go into the interlobal segmental arteries, go into the renal from the renal artery to segmental to interlobal to cortical radiate arteries. Arcuate vein deliver the blood to those cortical radiate vein will deliver the blood to arcuate vein. Arcuate vein empty into the interlobal vein. Interlobal directly into the renal vein. For some reason, we don't have segmental vein, like for the artery. It is empty interlobal vein empty directly into the renal vein, while the interlobal arteries get supply from the segmental artery, which get the supply from the renal artery. That's one lobe. Single. So we have that's the renal pyramid, right? That's one column over here, part of it. That's the adjacent for the adjacent lobe over here. That's the cortex, right? And all this part over here, that's the medulla, right? So that's the minor calyx. The interlobal vein empty directly in the renal vein. He got the blood from the arcuate vein. Arcuate vein got it from the cortical radiate vein. The interlobal arteries branch into the arcuate artery. Then into the cortical radiate artery. That's branch. I mean, we have these afferent arterioles that are branched from each, as you can see, each cortical radiate artery. We have those afferent arterioles that are branching from it. For the nerves, that for the blood supply, for the nerves, I mean, we have the renal nerves that innervate kidney and ureter. He enter each kidney at the hilum, all right, and he go innervate each individual nephron. The most innervation is sympathetic innervation that adjusts the rate of the urine formation by changing the flow and the blood pressure of the nephron. And most importantly, it stimulates the release of the renin, which restricts the losses of the water and salt in the urine by stimulating reabsorptions at the nephron. So this is this diagram is so beautiful because it summarizes really the blood vessel for the kidney. So we have the aorta that's branch into the renal artery, that's branch into the segmental artery, that go to the interlobal artery, arcuate arteries, cortical radiate arteries, and each cortical radiate artery branch into afferent arterioles that go to the capillary bed, which is going to be called the glomerulus here. And then he will go out from the efferent arteriole to the peritubular capillary, or what we call vesa recta, and then it will branch to it will empty to the cortical radiate vein, and from the cortical radiate vein it will empty to arcuate vein, interlobal vein, then the renal vein, then to the inferior vena cava. We are going to see this in detail soon, that the cortical radiate vein can also empty, the peritubular capillary can empty either to the cortical radiate vein or directly to arcuate vein. What is nephron? Nephron are the structural function units that form the urine. They are more than 1 million per kidney. They have two major parts, renal corpuscle and renal tubule. So this is the structures of this is sections of kidney. Apparently this is a right kidney. So we said that we take it bigger. So we have two parts. We have the renal corpuscle, right? Renal corpuscles is made of glomerular corpuscle and the glomerulus. The glomerulus is a type of capillaries composed of fenestrated endothelium. Highly porous capillaries. I don't want to say fenestrated. I will say a lot of porous capillaries that allow for efficient filtrate. Filtrate is, retain this word, filtrate is like kind of plasma but without protein. So doesn't have proteins on it. So filtrate, allow filtrate formations. And that's the glomerulus. The glomerular capsule, which is another part of the renal corpuscle, is also called Bowman's capsules. It's a cup shape. It is a cup shape. Glomerular capsules. It's a cup shape. So this is popular. It's capillaries with endothelium, fenestrated endothelium that are involved in allowing it's highly porous, that it allows efficient filtrate formation. And we have this glomerular capsules. Glomerular capsules, you have an epithelium that is squamous, actually. It's called Brown's capsules. It's like a cup shape, and it's consists actually of two layers over there. We have one that is outer layer. It's the parietal layer, which is just single squamous, as you can see over here, just single squamous epithelium. And we have, in contact with the glomerulus, we have this layer that is a visceral layer. Of course, it's contact with the inside, the visceral layer and we have this branching. It's made of a membrane. We have this basement membrane, all right? And this membrane, basement membrane, then we have this, of course, we have this fenestrated endothelium of the glomerulus. I told you that it's covered by these capillaries around it. We have this fenestrated endothelium of the glomerulus that will permit the filtrate to go in and out there. So what I am saying is that we have those parietal layer that is squamous. We have visceral layer that is it's close to the glomerular capillaries, and we have those branching epithelial podocytes, which are cells. And they are so funny, those cells, because those podocytes, if you see here the nucleus of those podocytes, they have those feet. Look at those. They are those feet. We call that a foot process, actually, that will attach those podocytes into the membrane basement. And between them, between those, you see those podocytes on those feet, we have those, see those things, this is what we call filtration slide between the process, allow the filter to pass into the capsular space. So to go to the capsular space, those not only the fenestrated endothelium of the glomerulus will allow that, but also those slight filtrate slides that they are between those feet that will allow also the filtrate to pass into the capsular space. Okay, so let's keep going. We have next we have the renal tubes, the renal tubules, those renal tubules. They are about 3 centimeters, 1.2 inch long. They consist of single layer of epithelial cells, but each region has its own unique histology and function. They have 3 major parts. We have the one that they are very close to the glomerulus. So this is called proximal convoluted tubules, close to renal capsule. We have the nephron loop. It's like a U, it's a loop, right? And we have the one that is far away from the capsule, renal capsules. It's called the distal convoluted tubules. And those distal convoluted tubules, they drain on this big, huge tube that we call collecting duct. So imagine the urine is made by the kidney. Look at what it go to before reaching the conducting duct. All right, and as I said, it consists of renal tubule. It consists of single layer of epithelial cells, but each region has their own unique histology. So let's look at this part, the proximal convoluted over here. Let's look at it in big. What we are seeing. We are seeing that colloidal, all right? This is an epithelium that is colloidal cells with a lot of microvilli. I'm making it bigger. with apical microvillosity. So we call proximal convoluted tubules. Sometimes we just put the initial PCT, okay? Retain that. And those villi, they form what we call the brush border, like in the intestine. Those villosity form also the brush border, and to increase the surface area for the intestine, to increase the surface area for absorptions, all right? This too, it increases the surface area and also have a large, a lot of mitochondria. The function, it's in reabsorptions and secretions, and it's very confined to the corpuscle, renal corpuscle. All right, and then we have the nephron loop. So let's look at the nephron loop, which is this part. This part is very funny because if we have, we call it also, oh, all right, so we have this nephron loop, we have the descending part and the ascending part. And look at the thickness. This is very thin, and then suddenly it's very thick when it becomes ascending. All right, it's very thick, the ascending, but thin also in some parts, but become very thick. And the epithelium is cuboidal or columnar cells over here, and is in contact with the distal convoluted tubules. The nephron loop, called also hand loop, this U shape, have also this descending lamp. Very thin. Then we have the distal convoluted tubule, or DCT, cuboidal epithelium, cuboidal cells over here, with the very few microvilli. He play a role more in reabsorptions than, he play a role in the secretions more than in the reabsorption. Then we arrive to the last one, which is the conducting duct. We have here, if you look at the histology, we have two types of cells. We have principal cells with very short microvilli. The function is to maintain water and sodium. And we have these intercalated cells. It's cuboidal cells with abundant microvilli. And this is to help acid-base balance of the blood. Very, very interesting. And these conducting ducts receive filter from so many nephron, and the row run through the medullary pyramid. And this is what gave the pyramid her their stripped appearance. The duct fuse together to deliver the urine through the papilla into the minor calyces. So the duct will come in together. Here I'm showing you just the minor calyx. They fuse together through the papilla into the minor calyx. So they are going to drain through the medulla. Again, the neural, this is an histology of cortical tissue, glomerular capsule, and this is the capsular space, and this is the papillary, glomerus. Very close to it, it's the proximal. Very distant is the distal. They have two major groups of the nephron, which is the structural and functional unit of the kidney. We have a cortical nephron that make 85% of all the nephron of the kidney, almost entirely in the cortex. So this is just the cortex, and this is the medulla, okay? And this is the cortex medulla junctions over here with the accurate arteries and accurate vein, all right? So the nephrons are going to be all in the cortex, right? The juxtamedullary nephron, he is very close to this junction, if you see it, and he invite the medulla. He go very deeply in the medulla. So is the conducting tubes. The ascending limb have thick and thin segment, and very important in the production of the concentrations urine. Glomerus, renal capsules, proximal PCT, nephron lobe, and then the distal convoluted tubules. What about their blood vessels and supply? Well, the renal tubules are associated with two capillary bed. We have the glomerus, all right? And we have also another capillary bed. We call it the peritubular capillaries. The peritubular capillaries. The juxtamedullary nephron, they are associated with vasa recta. accurate arteries, an accurate vein that's called, they are by the cortex medulla junction. So this is the medulla over here, and this is the cortex over here, okay? So let's look at about the capillary bed. The glomerulus, the capillaries are specialized for filtrations, right? They are, the endothelium is very fenestrated. And they differ from the other capillaries because they have a fed, they are fed and drained by arterioles. We have afferent arterioles that enter the glomerulus from the, that's the afferent arterioles, right? Afferent arterioles that branch I will go back and show you some very nice picture that we remember here. All right, so if you remember, those are the afferent arterioles that they are branching from the cortical radiate arteries. All right, segmental, accurate artery, interlobal, that came from the segmental artery. If you remember, we have the segmental, interlobal, accurate, cortical, afferent. Okay, so we go back. Afferent arteries enter glomerulus and leave via the efferent. So the afferent are coming in and leave via the efferent. Efferent. The efferent arteries, okay, so this is rising from the cortical radiate, right, artery. This is the cortical radiate artery over here. I'm looking at the red one, right? Efferent coming inside of the glomerulus, leave to the efferent arteries, arterioles, sorry. Which is going to be in contact with, you got it, those peritubular capillaries, the efferent. You see it with these purple ones over here. All right. So the supply came from cortical radiate artery into the afferent arterioles, enter the glomerulus, leave the by the glomerulus via the efferent arterioles, and go to those peritubular capillaries. So those peritubular capillaries, they arise from the efferent arterioles, and they cling to the adjacent renal tubules in the cortex. And those, if I follow them UP, they are going to empty where? They are going to empty into venules. There you go. Okay. The peritubular cavities, they are very porous capillaries adapted for absorptions for the water and solid, and they are low pressure, and they will tell you why it's low pressure. Inside of the glomerulus is high pressure. Let me show you why. I'm showing you this picture. So you see how efferent arterioles are very narrow and this is very large. So they have a lot of fluid coming into the glomerulus. So the pressure in the glomerulus is very high because those efferent to go out is so narrow, so small as compared as their diameter is so small. So it's going to be high pressure here because it doesn't go out, all of it. So it came in, poof, and then it go out very slowly. And since these peritubular capillaries are arise from these efferent arterioles, they are also going to be low pressure because they are not receiving a lot of blood coming from the glomerulus. Okay. So let's go back to our picture. We have this long 10-walled vessels parallel to the long nephron loop of the juxtaglomerular nephron. We don't find the peritubular capillaries, but we found what we call this vesa recta, those long vessels parallel to this U tube, U form U tube, tubule. Or the arrow rise from efferent arterioles serving the juxtamedullary nephron. So that's the efferent arterioles. And that's the efferent arterioles. Efferent, efferent, afferent. It's always big. If you look at the size, it's always big. The afferent is always larger. All right. So those vasa recta, they arise from efferent arterioles. All right. Serving the juxtamedullary nephron. And their function, actually, is to form concentrated urine. So the kidney produces about 180 liters of fluid processed daily, but only 1.5 liters of urine is formed. The kidney filter entire body plasma each day, 60 times each day. It consumes between 20 to 25% of oxygen used by the body are rest. The filtrate, that is basically blood with no minus protein, is produced by the glomerular filtrations, and if you remember, the glomerular filtrations have all this endothelium fenestrated and have this porous capillaries too. And urine is produced from the filtrate. Urine is less than 1% of the original filtrate. It can constitute less than 1% and contain metabolic waste and anything that's not needed. They have three major renal processes for urine formation and adjustment of the blood composition. We have glomerular filtrations, we have tubular reabsorptions, and we have tubular secretions, and we are going to see all these three phases. Glomerular filtration produce cell and protein-free filtrate. Tubular reabsorption selectively return 99% of the substrate from the filtrate to the blood in renal tubules and collecting duct. And the tubular secretion selectively move tubular secretion. It's what we saw here, green, and the red is what we saw here, glomerular filtrations that happen to the glomerulus, all right? And the reabsorptions in blue, and then the secretions that move substance from the blood to the filtrate in renal tubules and collecting duct. So let's get started. Glomerular filtrations. First stage is a passive process, does not need energy to happen. No metabolic energy is required. Hydrostatic compression forces fluid and solid through the filtrated membrane into the glomerular capsule. No reabsorptions happen here. Remember that the glomerular capillaries are covered by podocytes that form those feet, and between them we have this filtration slides, right? And it's a squamous parietal layer, the capsule. And we have efferent and afferent arterioles. And these glomerular capsules are fenestrated pore on it. And we have those podocytes, as I said, that attach, they have those feet processing that attach to the basement membrane. And between them we have this filtration slide. So this is main of the reason why the glomerular filtration is so passive. I mean, no metabolic energy is required. Then so we said that hydrostatic process pressure forces fluid through the filtration membrane. What does filtration membrane mean? Into glomerular capsules. What does filtration membrane mean? Filtration membrane, this is this porous membrane between the blood and the interior of the glomerular capsules. It allows water, small unit, to pass. Normally, no cells can pass. They contain three layers. We have the fenestrated endothelium. See, it's fenestrated. We have the basement membrane that is fused with the basal lamina of other layers, right? And we have those podocytes that they have those feet processing. And between them, we have this filtration slide. So this is why we call it filtration membrane, because of all those structures. So they allow molecules smaller than 3 nanometers to pass: water, glucose, amino acid, nitrogenous waste. Normal cells cannot let the glucose to pass. It's too big for plasma membrane to pass. It needs a transporter. But this filtration membrane let the glucose pass, so prevent loss of all the water to capsular space, and protein infiltrates indicate membrane problems if you find the proteins. Step 2, that's for the glomerular filtrations that happen because of this filtration membrane, this typical membrane that exists, this kind of modified membrane that exists around those glomerular. So we have tubular, step 2 is the tubular reabsorptions. Reabsorptions because most of the tubular contents return them to the blood. We have it's transepithelial process where most of the nutrients can be reabsorbed. Water and iron reabsorptions is normally, like sodium and water, they are hormonally regulated and adjusted. They cannot like being reabsorbed like that. They are hormonally regulated, and we are going to see how the water and the sodium get reabsorbed. They include both process, passive as well as active tubular reabsorptions. And the substance can allow 2 pathways: transcellular pathway, that the solid enter the apical membrane of the tubule cells, travel through the cytosol of the tubule cells. So it's transcellular. So it's via the cells, through the cells, and reach the basolateral membrane of the tubule cells. He exists basolaterally or exit through the endothelium of particular capillaries. We have also paracellular route. This one is between tubule cells, and it's water and some solids are going to pass by tight junctions. I mean, it's also, I mean, it's going to pass between the cells, but not within the cells. But this is very limited because of if we have some tight junctions over there. You will limit this movement, and which type of type junctions too. Those tight junctions are very leaky in the proximal nephron. So this paracellular solid led to the movement of water, calcium, magnesium, potassium, some sodium, mostly in the PCT, in the proximal tubules, because those tight junctions are they are leak. So I recapitulate how the intracellular route involve transport across the cytoplasm or the cytosol, and transport also across the basal membrane, often involve the lateral intercellular space, because membrane transporters ion into this space. And then from movement from the interstitial fluid into the capillary. For the paracellular route, it involves movement through leaky tight junctions, because they are very limited. If they have some tight junction there, it cannot happen easily. But leaky ones can do it, particularly in the PCT. And then movement from the interstitial fluid, the fluid between, the interstitial fluid is between tissues. All right, this is the interstitial fluid, that's my cell, that's the blood, into the capillaries. All right, so let's look at the reabsorption of the sodium, how it's happened. I don't know if I have some picture. Yeah. That's water. Sodium is an ion, right? Is most abundant in the filth, right? It's transport of the sodium across tubule via primary active transport. You use a pump, a sodium-potassium pump, into interstitial space. Sodium is then swept by bulk flow into peritubular capillaries. So it is an active transport that is pumping the sodium from outside inside, and taking out the potassium from inside to outside. When that happens, remember that all the membranes, they are like salted banana. The sodium is always very high outside and less inside, and when it go against the gradient of concentration, we call it active transport, right? So the sodium reabsorption by primary active transport provide energy. It creates an energy that's mean for reabsorbing most every other substance. Once it's activated, that we call it the primary active, he creates a secondary active transporter. And this electrochemical gradient that has been created by the basolateral surface will give a push need for transport of other solids. So other organic nutrients are reabsorbed as a kind of like co-transporting with the sodium, such as glucose, amino acids, some iron, and some vitamins too. The water reabsorption is very passive, because the movement of sodium and the other solids inside will create like concentrated gradient over here. So the water, he will go from lower concentration of solid to higher concentration of solid. So the water will be moving because it will be reabsorbed by osmosis. And this is helped by water-filled pore called aquaporins. The water are always present in the PCT, and they are inserted in the collecting duct only if this hormone ADH is present, antidiuretic hormone. At the basolateral membrane, the sodium is pumped into the interstitial space by the sodium-potassium pump. All right, and this is active. More sodium is downhill entered by the apical membrane. This will create a concentration over here, a lot of, because this will open also other solids to come in. Other solids are coming in, it's become a leak. I mean, glucose, amino acid, this will create gradient concentration. So the water will definitely move from reabsorbed by aquaporin. It increases the concentration of the solid that are left behind. For the lipid soluble substance, they diffuse by transcellular route or some iron and other calcium, for example, chlorine and urine, diffused by paracellular route. So this is our primary active transport, and this is our secondary active transport. You have to understand before I go to here that 90% of the renal reabsorptions, I mean, sorry, 65% of the sodium and water are reabsorbed, many iron, almost all the uric acid, almost half of the area later are going to be secreted back into the filter. They are going to be reabsorbed by the proximal convoluted tubules. And during the nephron loop, the water can leave, but not the solid. So I don't want you to look at the secretions yet. I want you just to look at in the blue ones. So we have 60% of the filtrate volume are reabsorbed, water, sodium, bicarbonate, and many other ions, glucose, amino acid, and other. And this is due to the passive first and second active. By the distal DCT. The PCT also regulates the absorption of sodium by the aldosterone and regulates the calcium by the parathyroid hormones. In the loop, in the ascending part, we have absorptions of the water. In this part is iron, calcium, sodium, calcium, and chlorine. And in the conducting duct, it's happen. The water is reabsorbed if we have the ADH. So this is the proximal CT. The substances that are reabsorbed are water, lipid soluble solute, urea, some iron, sodium. Primary, passive, and osmosis, and secondary active transport happen over here. Nephron loop, some ions. Distal ions. If you can see here, there are no water. Then the collecting duct. And this, to absorb the water, we need ADH is required to insert the water through the aquaporin. Now we move to the third step, which is the tubular secretion. Is the reverse of the reabsorptions happen mostly in the proximal CT. And selected substances are going to be moved from this peritubular capillaries through the tubule cells into filtrate. Some of those substances are potassiums, protons, creatinine, organic acid, and base. And this is very important, the secretions, because such as drug metabolites are found into the plasma membrane and eliminate indesirable substances that were passively reabsorbed, for example, urea and uric acid, and get rid of the excess of potassium due to the aldosterone effect, and control the blood pH by altering the amount of proton or the bicarbonate. If you look at it, we have an acid and we have a bicarbonate, which is like a buffer over here in the urine. So the green one are all secretions. They happen most of it, as I said, in the PCT. Ureters arise from the renal pelvis. It's a long tube with the smooth muscle propel urine from kidney to urinary bladder. It enters the urinary bladder in the posterior inferior wall. There are three layers. You can see mucosa, musculus, and adventitia. Adventitia, it's outer fibrous connective tissue. The muscularis are mostly smooth muscles that contract in response to stretch. And the mucosa, it's very typical mucosa here. It's transactional epithelium. The muscularis, the smooth muscles, when they contract, they propel the urine into the bladder. Gravity alone is not enough, not even needed. It must also be pushed by a peristatic wave action of smooth muscles. And the rate of this movement, this peristalsis, adjusts actually the rate of the urine formation. And also it's under the parasympathetic and the sympathetic innervations. Urinary bladder, you have here a female urinary bladder. It's a hollow organ with a muscular wall that stores urine. A fold of mucous membrane over the opening that prevents the reflex. We have two arterial orifices. We have, those two over here, that the erythritic orifice, this is where the ureters are coming in contact with the bladders. And we have this orifice that is made of the internal urethral sphincter. So this made like a triangle over here. We call that trigon. The muscular layer, we have the mucosa, it's transitional epithelium mucosa. And the muscular layer is reserve muscles that contain three layers of smooth muscle, inner and outer longitudinal, with some middle layer. And the adventitia is a fibrous connective tissue. And it's connected with the urethra through an urethral sphincter. The female urethra is tightly bound to the anterior vaginal wall, and the external urethral orifice is anterior to the vaginal opening, posterior to the clitoris. The male urethra carries semen and urine in the same time. The three named regions, we have the prostatic urethra by the prostate. We have the intermediate part of the urethra past the urogenital diaphragm from the prostate to the penis. And we have the spongy urethra that he opened via the external urethral orifice. The urine is a aqueous solution of greater than 90% more of water. You have urea. He sensitizes the liver from oxidations of amino acid or from ammonia. Those urea. You have also chloride, sodium, potassium, creatine, other ion, inorganic and organic compound. So those, all of them have to be in physiological dose, because more of some will tell you that your kidney is not functioning correctly. Involuntary PP, I mean urinations that you cannot avoid, micturations. We have a three simultaneous events that may occur: contractions of the this muscle, the detrusor. By the detrusor is the muscle, if you remember, that it's around the urinary bladder. Opening the antral urethra sphincter by the autonomous nerve system, or opening of the external urethra sphincter by somatic nerve system. In the kids, distensions of the bladder activate stress receptors, causes excitation of the parasympathetic neuron and reflex center in sacral region of the spinal cord, leads to contractions of this muscle, the detrusor, and opening, contractions of the internal sphincter, and the inhibition of the somatic pathway to external sphincter allow its relaxations and openings, so therefore the P came out. Putting control center mature between the age of 2 and 3 years old. Putting storage center inhibit micturations, this avoiding urinations. Inhibit the parasympathetic pathway, excited sympathetic and somatic efferent pathway, and putting micturition center promote micturations, excited parasympathetic pathway, and inhibit the sympathetic and somatic efferent pathway. And we're done. Thank you so much. I will see you for the next